So here I will enlarge the axon terminal right there. And then I will draw the skeletal muscle fiber side. Okay, so what I'm going to explain here is what happens at the NMJ, or so-called neuromuscular junction. So for neuromuscular junctions, it is connection between your motor neuron, in that case, that will be equivalent to the postsynaptic neuron right here. This particular neuron can form a synapse at the end of here with a skeletal muscle fiber, and that's what I'm depicting here. So the motor neuron now becomes, in a sense, a presynaptic neuron that's going to release acetylcholine to the skeletal muscle fiber side, because the skeletal muscle fiber is our effector tissue that we're trying to tell it, hey, muscle, you got to contract. All because the whole purpose of this action potential in a neuron is just to tell what to do, things down the road, what to do. Whether it be the effector be a smooth muscle, cardiac muscle, skeletal muscle, you always need to have an action potential in order for you to, from the brain, to tell a particular part of the body to do something as a response. And that's really what why action potential is so critical. On the other hand, if we're just talking about how skeletal muscle fiber contract, you have the neuromuscular junction, where this is the motor neuron, and on this side, you have the skeletal muscle fiber. So this is an enlarged axon terminal that we saw in the previous page here, okay? So action potential, I'm going to sort of draw it as a little red arrow like this. Okay, let's say an action potential has been fired due to all these events happening, going through the telodendria and reaching the axon terminal. Now, right before the axon terminal, you have another channel, it turns out. And that channel right here is what we call, maybe I'll use this color to outline it so I can write it, what it is. This is a voltage gated calcium ion channel, okay? And this is for the purpose of allowing calcium ion to come in to the axon terminal. And what calcium ion being inside the neuron's axon terminal do is it induces these, remember these are little, what we call synaptic vesicle. And inside we have neurotransmitter. And in this case, because we're talking about telling the skeletal muscle fiber to contract as a response, these little dots here represents the neurotransmitter acetylcholine, which we abbreviated as ACH. Now, these neurotransmitter are already made and they're contained within the synaptic vesicles, and they will do nothing unless they see calcium. So this incoming calcium from the voltage-gated calcium channel is to induce exocytosis of these acetylcholine to the synaptic cleft. So this right here is what we call the synaptic cleft, or the little space between the axon terminal and the skeletal muscle fiber. Because when we're talking about connection between whether it be between neuron and a neuron, or a neuron and a muscle fiber, there's always no physical touching or connection between the two. It's just sort of separated by a gap such as this that we call the synaptic cleft. Okay. So calcium ions will cause exocytosis. So I'm going to draw it kind of like that, where the vesicle will slowly travel towards the membrane of the axon terminal to allow for exit of these acetylcholine into the synaptic cleft. Now, in this particular spot, I'm drawing this curvature where the axon terminal forms a synapse with the skeletal muscle fiber. just as we call this axon terminal, this little indentation for the axon terminals to sit in, we also give it a name. So on the skeletal muscle fiber side, we call this part your motor end plate. This is where it really have this literal indentation for these axon terminal to be forming a synapse and then sitting in there, like a little finger sitting on top of a tube, okay, if you will. But so this is a synaptic cleft where the motor end plate is. And so this little gap here is where the neurotransmitter acetylcholine will go into. Now in there, now on the skeletal muscle fiber side, on the motor end plate, you also again have sodium ion channel. And these were essentially the same ones we saw here, okay? But now it's just on the skeletal muscle fiber side instead. So these are what we call the chemically gated sodium ion channel. Okay, remember these are chemically gated because there's no hopping over of this action potential. So this little red line here, I will re-emphasize that this represents the action potential being fired from the motor neuron itself. There's no way for this action potential to hop over to the skeletal muscle fiber. No such thing can happen. There needs to be something else that starts an action potential on the skeletal muscle fiber side. Because essentially, what is an action potential for is to relay that signal to the next, either neuron or the muscle fiber or effector tissue in line. So this action potential has to release acetylcholine. And the purpose of releasing acetylcholine in the cleft here is to activate another round of action potential down into the skeletal muscle fiber side. So that's why this acetylcholine will actually bind to those chemically gated sodium channels, which opens them and allows for inward movements of sodium ions. Now remember, just like before, there's no sodium ion being released from the axon terminal. These sodium ions are just coming in from the interstitial space between the neuron and the muscle fiber. Okay, so that's why they're all over. All of this space here is filled with sodium ions, and they're just flowing in from the synaptic cleft into the skeletal muscle fiber. So just like how that your influx of sodium ions caused an action potential to be fired on the neuron side, same thing happens for the skeletal muscle fiber, but we're not going to go into the details of what happens as to how that gets started, but it is a similar process where this influx of sodium ions triggers an action potential. Okay, remember this red little arrow thing? I'm just going to continuously use it as a symbol of action potential being fired. Action potential. But now we're talking about action potential moving across the entire skeletal muscle fiber via the sarcolemma. Okay. So sarcolemma, it's really the plasma membrane of the skeletal muscle fiber. So this action potential will be spread across the entire skeletal muscle fiber via their plasma membrane, which is called sarcolemma. And once it spreads across the entire skeletal muscle fiber, it will go down these little tubes that goes into the muscle fiber. These are, you can imagine these as little wires that wraps around the muscle fiber in big tubes, big hollow tubes. We actually call these t-tubule, or also, stands for transverse tubule, because it kind of cuts the skeletal muscle fiber transversely like that. And between two t-tubule, we have another structure right here that kind of looks like a little meshy little thing where there's holes in between, and the little expanded region like that towards the end where it touches the t-tubule, okay? This whole thing is what we call the SR, or sarcoplasmic reticulum. And then within the sarcoplasmic reticulum, we have storage of a lot of calcium ions. Lots of calcium ions within the SR itself. That's the whole purpose of SR within the skeletal muscle fiber for. Now, once we have action potential firing across the sarcolemma on the skeletal muscle fiber side, it now travels down into the T-tubule. It has to travel into the T-tubule, and this action potential firing in the T-tubule, T-tubule is what will stimulate the SR to release the calcium. So with this little section and this section here, sort of widened section of the SR, is what we call the terminal cisternae of the SR, okay? So either this side or this side, it is called terminal cisternae of the SR, where its main job is once it sees action potential being fired down the T-tubule, calcium ions will be released into the sarcoplasm. Remember, this whole thing is the skeletal muscle fiber, and skeletal muscle fiber means that it is one cell. So within the cell, you have the cytoplasm, as we all know, and the skeletal muscle fiber, this particular cell also have a cytoplasm called the sarcoplasm, which is all over within this cell right here. It's very long. Remember, skeletal muscle fibers are very long, so I can't draw the ends of the skeletal muscle fiber because it can go on this way or on that way. But just remember that all of this space is really sarcoplasm, where the organelles are around, and now you have calcium coming out of the SR for a different purpose, okay? So make sure you don't mix up the calcium ion that's happening here versus the calcium ions that's happening here. Remember, calcium ion that is released, or where it is going through the voltage gated calcium channel, is only for the purpose of initiating exocytosis of acetylcholine. On the other hand, calcium ions released by the SR is what's going to trigger a muscle contraction by the sliding filament theory. I will explain what that is next. So once calcium ion now is released into the sarcoplasm, so what's going to happen is I'm going to kind of draw it on the side here versus on another page. That way we know it's all connected. Okay, so just imagine in this little panel, we're really zooming in to what's happening here, because what we have down here is really the sarcolemma, the sarcoplasm that has your sarcomere, which are individual functional unit of a skeletal muscle fiber. Okay, again, I don't have time to talk about all the parts of what a sarcomere is made up of. We may talk about that in another video. But the whole purpose of calcium ion is to bind to something called troponin. Okay, so remember, in the muscle fiber, you have thin filament and thick filament. Thin filament is what we call the actin filament. So I'm going to draw the actin filament's main characters. So it has a lot of little spheres like this that kind of winds around a central line. Okay, and that central line is what we call the nebulin. Whereas these little circles here are what we call g-actin. So all of this is part of one actin filament. G-actin has specific dots or spots on them that we call them little active sites on these G-actin. And these active sites is for myosin head to bind. Okay, we'll talk about what that is in a bit. And then on top of that, you have a little molecule that lays right over all the active site, that molecule that winds around the g-actin and covers the active site perfectly, this molecule is called tropomyosin. Okay, tropomyosin. And then tropomyosin can be controlled by another protein called troponin. And I'm just going to draw that on the surface like this. Here, like that. Okay, it's like a little clip that has 3 subunits. So this guy right here is troponin. So remember, calcium has just went into the sarcoplasm and it's now enlarged into this particular actin strand. So now calcium ions are all around the sarcoplasm like that. It actually then binds to troponin. Troponin, its job is to bind to calcium, and once calcium is bind, it will move tropomyosin away, okay, and reveal the active site of g-actin. There's no animation here, so I can't show you this. But what happens is once calcium binds to troponin, troponin actually rolls or kind of roll it or move it to reveal, it moves the tropomyosin so that it reveals all the little green dots underneath on the g-actin, so that once the green dots, let's say, is revealed with the active site being available, you have the little thick filament and its myosin head that can bind to form what we call a cross bridge. Okay, so this is myosin, and that's the myosin head. And when myosin head now sees the availability of the active site on g-actin, that's what we call the formation of a cross bridge. Okay, and once a cross bridge is formed, the little myosin head will actually move like this. Okay, it will move and pull the actin this way. Okay, so it does what we call a power stroke while still connected. Imagine that this is the little g-actin going that way. Okay, so you have the g-actin now here, okay, and it's gonna move inwards when the myosin head kind of pulls like this. So if I were to illustrate it with my little finger, this is the myosin head. Here's the actin binding, you know, where we have the g-actin on here with the active site. The little myosin head now sees the availability of the active site. It will bind, and then it will pull the actin inwards in what we call a power stroke. Okay, so before it is where the myosin, let's say, is, the actin is rolled, where you don't have the availability of the active site on g-actin. Once troponin binds to calcium, it reveals the active site, allowing the little myosin head now to bind, forming a cross bridge, and then it will pull the actin inwards so that it moves all the actin towards the M line, which is in an action called the power stroke. And that's how you're able to produce a muscle contraction at the end of the day, okay? Where you have, remember, all the action potential happening down the length of the neuron, and then you have what's happening at the NMJ, neuromuscular junction, and excitation contraction coupling happens in here, where we have action potential moving across sarcolemma down the t-tubule, causing calcium ion to be released from the SR into the sarcoplasm, which that calcium then can bind to troponin. Troponin moves tropomyosin out of the way to reveal the active site on the g-actin, allowing the myosin head to bind, which then does, well, forming a cross bridge, and then it will automatically form a power stroke, which pulls the actin inwards to form a muscle contraction, kind of like this kind of motion. I kind of see, you usually use my fingers to illustrate that as a way to say, if this is the actin, they're coming in like this in a muscle contraction like manner. Now, what can release the myosin head from the g-actin is because it got to be released in order to keep doing this over and over again. This is where ATP then have to come in. Okay, so I'm going to use a little star to represent ATP. Okay, so this is where ATP comes in, and ATP's purpose is to release the myosin head from the g-actin, allowing it to go back to its original position and bind to another g-actin at another active site. So that's what ATP is necessary for in terms of the troponin and tropomyosin actin myosin type of connection. It is to break or release the myosin head from the actin. That's where one location where ATP is needed. Another location where ATP is needed, I'm not going to draw it in there to not confuse you, is when the muscle needs to relax other than myosin head needs to be released from the actin. All these calcium in the sarcoplasm also have to be pumped back into the SR. So because it's going, against this concentration gradient, it have to be using ATP to pump all the calcium ions back into the sarcoplasmic reticulum so it can be all stored back in there and ready for the next action potential to come through on the skeletal muscle fiber side. And that's really all for action potential in a neuron, as well as what happens in the muscle fiber side in order to generate a skeletal muscle fiber contraction.